All right, so the question was, do we have a date for the exam? As of right now, it still looks like it's going to be a week from today. It'll be like usual, online. Yeah, so unless I get a bunch of emails where someone has some legitimate reasons why they don't want it on Friday, then that's probably what's going to happen. Most likely we'll finish the chapter that we're working on right now, the last chapter, chapter 10. We'll probably finish that today or tomorrow or Monday. So that means my idea is you'll have a lot of time to study for it. Meaning, if we're doing the exam on Friday, which again is the current plan, hopefully we'll do a non-mandatory meeting online for an exam review, probably Wednesday or Thursday. I guess Wednesday would be better because, the exam review, you get all your answers that you have. So that way you'll have a full day to study. So we'll see. Good morning, Fitzpatrick.
Good morning.
Good morning, Baumgartner.
Morning, sir.
Got less than a minute left. So I'll go ahead and tell you guys online then. I'm just going to do the same thing I've been doing. It seems like a good system. If you're here and paying attention, you just have to send me the words for attendance. And if I have time to make questions, then anybody who didn't get those words in by then will just have to do the questions. I'll give you a second, but I'm about to give you the first word for today here in a second. Actually, I'll do it right after attendance, so you'll know when it's coming. Really quick, I'm going to do some announcements. The Evolution Lab, as always, is due on Tuesday, right? Because you have a week to do it. So just reminding you that that's due. If you have questions about it, let me know. Speaking of lab, I realize I haven't given you your lab for next week. So yeah, there is a pre-lab. There might be a pre-lab on it, but since it's already Friday, what I might do is incorporate the pre-lab into the lab because I don't think it's necessarily fair that I give you, a certain amount of time to do the pre-lab and then I give it to you late. So most likely, I'm gonna try to figure a way to incorporate that pre-lab into the actual lab. And I still need to look at it and decide if lab's going to be in person or online. I want to do online again because moving forward, nothing we do in class is going to involve hands-on experiments, right? Because we're looking at huge things, evolution, ecology, things like that. So those are things you can't simulate, right? We can't make mice breed and then have, 10 generations and look at how genetics is passed down and things like that. So most likely, moving forward, everything will at least be on the computer. Now, whether it's online, do it on the computer on your own or meet here to do it, we'll see. But be prepared for that. Any questions about lab announcements? That's all the announcements I have. Let's do attendance very quickly. If you're coming in late, I did answer a question about finals, but you can ask, not finals, about the next exam, but you can ask people about that, or you can watch the video, or you can talk to me during office hours. So Cooper is here. Breeden is not here. Elias, not here. Hamrick, not here. Lloyd is online. Davis is online. Ray is online. O'Brien is here. Michael Baumgartner is online. Pena is not here. Sigmund is not here. Johnson, not here. Bayard, not here. Huffman, not here. Hamlet is online. Thornabar is here. Fitzpatrick is online. Tyrus is here. Lacey is online. Sullivan is not here. Hill is not here. And Byrne is not here. Did I miss anybody? Okay. So like I already said earlier, for attendance for you people online, we're just going to do the words for now, just like we've been doing for the past week. So the first word for today is jack, like the name Jack, or a floor jack, or a car jack, whatever. The word jack is the first word. Okay, let's jump back into chapter 10. We ended talking about DNA replication, right? So what we ended with was telling you the results of DNA replication. And this is a very important idea here, something that you're going to need to know for the exam. So first of all, remember, what we're looking at here is DNA replication. And to remind you what we talked about in this chapter and chapter 8, this happens in the S phase of interphase, right? Most of you guys got that correct, or seem to understand that concept when we were talking about mitosis, so I think you'll do good. This happens in S phase of interphase. The other thing you need to notice here is that we start with one DNA strand. I'm just going to put mom, like that's the mother strand, and I'm using this terminology. This isn't from your book. I'm going to call that the mother strand, right? And that strand has to duplicate itself in S phase of interphase because it's getting ready for mitosis, which is where the sister chromatids are going to split, right? So it has to duplicate everything. So that's what it's doing. It's duplicating its DNA. This strand of DNA is being duplicated. Excuse me, this double helix of DNA. This is where I need to be very careful. This blue part, right, that we're looking at here, that is a double helix of DNA. A double helix, as we learned on Monday, consists of two strands, right? Here's strand 1. Here's strand 2. I don't know if you guys can see that online, but if not, I apologize. Strand 1, strand 2, right? That makes up the double helix. Now, what happens in S phase of interphase is those strands are opened up. Because as I made a big point to discuss on Wednesday, the thing holding those two strands together is the weakest of the bonds. It's hydrogen bond. So it's nice and easy to pull apart. So once it's pulled apart, then those, here we go, these things right here, right? These bases are the template. So there's cellular machinery that reads the template. Like, oh, there's an A, I better bring in a T. Then there's a C, I better bring in a G. There's a G, I better bring in the C. That's what's happening, right? More importantly, what we need to look at here, and this is a question on your study guide. We start with this one double helix, right? The mom strand, or the mom double helix is what I'm going to call it, and then it involves two strands. And then at the end, we have two new double helices, right? And I'm going to call these the daughter. And again, this is just my terminology. So don't take notes. I'm not going to call it that in the exam. I'm just trying to make an example here. So now we have two new daughter, a double helix, right? Double helix here, double helix there. That's the new stuff. Now the question in the study guide and probably on the exam is, what is each new double helix consist of? And the answer is that it consists of one old strand, right? You can see that blue one, that's the old strand, and one new strand, that's the gold, right? And you can see why, right? Again, it's the bases that are the template. So you open it up. You open up that old double helix and read the bases. So if there's no questions about that, let's talk about how this actually happens. Are there any questions about that? All right. So this next slide I'm going to show you is a bunch of words that basically I've already said, but it's in writing. So here you go. DNA replication, again, it happens in the S phase of interphase. Here's the new stuff, though. And I might ask you this middle bullet point here. Where does DNA replication begin? It begins at the origins of replication. So you should know that word, at the very least, because I might use it again this chapter. Origins of replication, that's where it starts. which is good because it's in the name, right? Origins of replication. And I think that's slightly important because you might picture, if you look on the left, I'm going to draw a string of DNA. Assume that's a double helix. I'm not going to try to draw a double helix. You've got one end, you've got the other end. I think intuitively, some people might just assume DNA replication starts at one end and then goes down until it gets to the other, right? That might be the intuitive way to think about it. So I'm glad your book points this out because that is not how it happens. What happens is you have these origins of replication and you have quite a few of them. So your book is zooming in into a double helix right here. That's what this picture is right here. It's a double helix. And you can see the normal parental strand right here. And then you can see, because we've zoomed in, and we're looking at a very small section of a huge chromosome, there's two origins of replication. That's just an example. In real life, there's going to be many, 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 many origins of replication on your entire chromosome. And the cellular machinery that we're going to talk about later, it attaches on right there, the origins of replication. And then it starts doing its business. And it gets to work. Whoa, excuse me. Yeah. And that's what happens. And then what happens is duplication moves in both ways, right? So it's going to go this way. It's going to go this way. This one's going to go this way. This one's going to go that way. That's not a detail you need to know for the exam. That's just for your own knowledge. So yeah, it keeps going. And if you could picture it, imagine this bubble on the left increasing, right, as it moves to the right. And this bubble increasing as it moves to the left until finally it's all one, right, when it's together. And that's when you end up with this, which is basically a different way of looking at this last slide. Come on. Yeah. Come on. Sorry, my computer's slow. This slide right here, you can see the blue representing the old strand and the gold representing the new strand. It's the same as this. But anyway, for the exam, the important thing here is that DNA replication happens in the S phase of interphase and that it starts at the origins of replication. If we were still doing independent work, that would be a good topic. I might ask you how many origins of replication are on each chromosome, or if it's the same. Like if my chromosome number 7 has 1,000 origins of replication, does every other human have 1,000 origins of replication? But anyway, any questions about this slide? All right, if you download the PowerPoint, in this case, I really recommend it, or do a Google search if you want and find a video, or maybe I'll do a Google search and find a better video and share it with you. But you should really watch this animation. It really shows what I was trying to show. Sometimes a picture really does the trick. Sometimes an animation really is needed. In my opinion, animation really is needed. So check that out if you want. Same here. There's another video that shows the same thing. As with any of these videos that I show, if there's any words that you don't recognize, like in this case, that one's titled leading strand, and the next video is titled lagging strand. And that's important for a biology major. But for you guys, you don't need to worry about those numbers or those names. Same thing here. 5 prime, 3 prime, nothing. Don't worry about it. If I didn't give you that information, there's nothing to worry about. But anyway, here's that machine that does the DNA replication, and we'll talk about that here shortly. So any questions so far? All right. Now we are on to our second main bullet point. So we're done with the first one. Hooray. The first one was a little bit easier. The first bullet point that we just finished, it's a little bit easier than the second one because the first one was a lot of review, right? Because you already knew a lot about the structure of DNA thanks to chapter 3. Now we're getting into some newer information. And in my opinion, this one bullet point might be one of the most important bullet points of the semester, given that we're in the middle of a pandemic. So if you understand how mRNA vaccines work, you need to at least know this first. If you don't know this and you think you understand how an mRNA vaccine works, you don't. You're relying on somebody else, which don't get me started on that, but everyone will call you a sheep. And I say everyone's a sheep, and that's okay, because you're either following an expert who knows this stuff, or you're following someone who's not an expert. Either way, unless you know this stuff, you are taking information from someone else and trusting them. So here we go. Here's some information for you. If you want, you can do your own research. And I use quotes because it's not a research, it's more like a literature review. But let's get started. As you probably already know, DNA provides instructions, right? It's a set of instructions. Like I said in, I think it was chapter 8 and chapter 10, I used the analogy of your chromosomes like just a book of instructions on how to build a protein, right? That's instructions. That's what DNA does. So it provides instructions to a cell, because your cell, I've used this analogy before too, it's almost like a factory. It needs to build things. First of all, just to maintain itself, right? So that's why it would provide instructions for a cell. And also because a cell is almost like a factory, the factory also provides, it builds stuff not just to maintain itself, right? It also builds products that it's going to export. Same with a cell, right? The cell builds things that it needs just to stay alive, but also your body needs certain things from certain cells. So your cells produce those, right? So cells in your liver might produce bile, for example. Cells in your bone marrow are going to produce white blood cells, things like that. As you already know from the previous chapters, the genotype is the heritable information, right? So your chromosomes that you got from your mom and your dad, that is the heritable information. It is the sequence of nucleotides in the bases of the DNA, right? So this is slightly new information. You already know that the genotype is the heritable information. And I've already told you what I'm about to tell you, but this is the first time I think you've seen it in writing. It is the A's and the T's and the C's and the G's. That is the code, right? When I say there's information stored in DNA, that's it. It's the bases. It's not the sugars, because all the sugars are the same. It's not the phosphates, because all the phosphates are the same. Just like if we go back to the analogy of the chromosome being an instruction, a book of instructions. If you were looking at a book of instructions, it's the letters that provide the instructions, right? It's the alphabet, A through Z. It's those words that they put together to tell you, all right, first get this piece of lumber and attach it to this piece of lumber using this bolt. Now take this piece of lumber, right? You follow those instructions written in English. And those are the words. That's the code. It's not the blank spaces between the letters. It's the letters. I know that seems like I'm beating a dead horse, especially for such a simple concept, but I want to make sure you understand it. So when we say DNA provides instructions and it's a code, it's the letters, right? That's the information. And as you already know, this part is a review. The phenotype of an individual, right? That comes from the actions of a wide variety of proteins. So basically, these two bullet points right here, they say the same thing you've already learned, but now we're just saying it in a different way in the context of a chapter that's all about DNA. So the codes in your DNA make up your genotype, right? Those letters make up your genotype. And your genotype makes up your phenotype. So then the question we should ask ourselves is, how does the DNA provide these instructions? So here we go. That's what we're going to talk about. How an organism's genotype determines its phenotype. And the good news is I've been giving you a little bit of this information. So when we were talking about this in chapter 9, even though your book didn't spell it out yet, I was already hinting at it. So some of this should come as a review. First of all, as you already know, and obviously as you need to know, because we've used this word in more than one chapter now. A phenotype, according to your book, are the physical traits from the actions of a wide variety of proteins. I kind of like the last chapter's definition better, which is basically your phenotype is the result of your genotype, right? The reason I don't like this definition is because it says physical traits, and that's true. But a lot of times when people think of physical traits, their mind goes back to that lab. Dimples, eye color, hitchhiker's thumb, things like that. But sometimes the physical trait could be a disease, like when we talked about achondroplasia or sickle cell anemia or cystic fibrosis, right? The physical trait doesn't necessarily have to be something you can see. And your book gives some examples. If your hand's tired, I wouldn't write these down because these are just examples. I'm not going to ask you on them, but some examples of this are the structural proteins that help make up the body of an organism. So that's straight up, right? So straight up, if your genotype says make this protein, not that protein, that would be this bullet point. And here's where it's actually more realistic, is this second bullet point. What often happens is that the code in your DNA and your genotype says, all right, build this protein, just like it did in this first bullet point, except this time that protein that it's making is an enzyme. which makes sense because let's think about it like this. Chin dimples. Remember when we talked about that? That's on your chin. If you had to guess, let me think. How am I going to say this the right way? I'm trying to, let me think of a better example. Eye color, hair color. Okay. Hair color. I won't get into that either. I don't want to make this confusing. I'll keep it simple. Let's not do specific examples. Think about it like this. Not everything that you see that makes you different from somebody else is directly because of a protein, right? Sometimes it's because of an enzyme. All right, here's a good example. It's not for humans, it's for plants. So when we talked about Mendel and his pea plants, they were either white flowered or purple flowered. You guys remember that? And it wasn't the thing that made the pigment wasn't a protein. It was a pigment. It wasn't a protein. But it was an enzyme that helped catalyze that reaction to make the protein. Does that make sense? So what happens in this case is your genotype might code for a protein, and that protein is an enzyme. And that enzyme is the machine that makes that pigment for that flower. So it isn't necessarily that that gene itself says, here's how you make the pigment. It was more like that gene said, here's how you make the machine that makes that protein, that pigment. Of course, I'm talking about all that, and really, I'm not going to go into detail on the exam. The most important thing on this slide is that you know that the phenotype arises from the genotype. And the phenotype is essentially the physical traits. Just remember, physical traits does not always mean things you can see. Any questions about this? The next slide is going to be a little bit more important, and it will be new information. The second word for today for the attendance is Johnson, like the name, usually a last name, Johnson. All right, next slide. DNA specifies protein synthesis in 2 stages. So far, I've already told you that DNA specifies protein synthesis, right? I keep saying DNA provides instructions on how to build a protein. It does this in 2 stages, and you need to know the 2 stages. You can write them down now if you want, or you can wait, because I'm certainly not going to leave it at this. We're going to talk about both stages in detail. One of them is transcription, and the next one is translation. They are sequential events. First, transcription has to happen, then translation has to happen. What is transcription? That's when you transfer the information from DNA to RNA. And translation is when you take that information that's in the RNA and you actually translate it into a protein. Everybody remembers things differently and studies differently, but I recommend studying this chapter like I recommended studying photosynthesis and respiration. Start with the basics, memorize the basics, and then start adding in details. Write it as a sentence if you want. Your first sentence should say, DNA provides instructions on how to build proteins. Once you have that memorized, then add this other part in. DNA provides instructions on how to build protein in 2 stages. And once you have that memorized, DNA provides instructions on how to build proteins in 2 stages, transcription, then translation. And then once you have that memorized, then say DNA specifies or provides instructions on how to build protein in 2 stages, transcription, which is the transfer of information from RNA to DNA, and translation, which is going from RNA to polypeptides. And sorry, I slowed down there because I said a wrong thing. I'm gonna make sure I correct myself. In transcription, you go from DNA to RNA, not the other way around. In the following slides I'm going to show you, we're going to talk about all these things. We're going to go into detail. But before I do that, I want to go back to my analogy. If you think about the cell as a factory, right? And in the middle, and your factory builds a bunch of stuff. Like I already said earlier, sometimes that factory builds things just to maintain its own factory, right? Oh, it needs light bulbs. It needs power outlets, whatever. It's building stuff just to maintain itself. But also it's a factory, so it's also building things to export. Just like a cell is building things it needs to maintain itself, but also sometimes a cell will build things it needs to export for your body. So in this analogy, your factory has one building or a room in the middle, right? And in that room, you have all these books of instructions on how to build things. You guys are with me so far, right? It's a library, if you will, with a set of instructions on how to build things. But here's the thing. This is prior to the internet and all that. You only have that one set of instructions, right? You've got the one book for chromosome 1. Well, technically you have 2, right? One for your mom, one for your dad. But that's it. You only have those copies. So let's say somebody from the factory floor says, hey, I need to build this thing. He walks up to the library. He knocks on the door, says, hey, I need instructions on how to build X, Y, and Z. What do you think would make sense? Should we give that construction worker the whole book, the only copy you have, and said, all right, take this book and this is how you go build it? Would you do that? Why not?
Because you will lose information.
Yeah, you run the risk of that construction worker losing it, or maybe accidentally dribbling some dip on it, or maybe spilling his coffee on there, or God forbid, beer. You'll fire him for that. But yeah, you don't want to give your construction worker the only copy that you have, right? Because you need that only copy. So what would you do then in that case? If the construction worker is asking you for instructions on how to build X, Y, and Z, what would you do? You make a copy, right? Well, let's assume there's no copy machine.
Give them instructions.
Yeah, so you would write it down, wouldn't you? If that's all you had the books, and you also had some loose leaf paper and a pencil, you would read it and you would write it down, right? And let's say those books are written in English. Would you, and your maintenance worker or whatever, he asked for it in English. Hey, man, can you give me some instructions? It didn't say it in Espanol. He said it in English. So when you write it, are you going to go from English to English? Are you going to go from English to Spanish? What are you going to do?
Give it to him in Spanish.
Well, no, I'm saying he spoke English.
Oh, he spoke
so, yeah.
Give it to him in English.
Yeah, right? So you're not translating anything. You're just going from English to English. The only thing is you don't want to give them your only copy. And that's what happens in your cells. Your DNA is your only copy. So when something needs to be built in your cells, somebody's knocking on your nucleus saying, hey, I need instructions on how to build X, Y, and Z. Your nucleus is like, all right, hold on. Let me write it down and give you the instructions. And that is what transcription is. And it's in the name. Hopefully you guys have heard the word transcribe before you've had this class. Because if you think of that word outside of biology, it means to write something down. Right? So in this analogy, we're just taking those instructions and we're writing them down because we don't want to give out the only copy. Now, the construction worker has these instructions, right? He goes to wherever his workbench is, or she is going to make whatever product it is. At that point, the construction worker, when you think about it, is really doing a translation, right? He's taking, or she is taking those written words, those English words written on the paper, and translating that into a 3-dimensional object that he has to build, or she, right? All right, so that's the analogy. Keep that analogy in the back of your head because that's exactly what happens with cells. Here we go. First, we're going to do an overview, just like we did with photosynthesis and respiration, right? That should give you, it's a bad omen. That should tell you this is probably going to be a little bit complicated. The information in your DNA, like we said, is transcribed to RNA. So going back to the analogy, what we're saying is the information in your only book of instructions, it's copied, it's handwritten. And that handwritten message, that's the RNA. So whatever piece of paper that guy wrote down on, that is now the RNA. And then that RNA is then translated to the polypeptides. And then that folds into proteins. Now, this is the thing we're going to talk about later. The relationship between polypeptides and proteins. For right now, you can think of those as the same words. They are different. We'll talk about why later. But for this point in the lecture, you can think of those as the same thing. So again, DNA is transcribed into RNA, and then that information in the RNA is translated into proteins. Here's a picture of what I'm saying. There's your DNA, aka your book of instructions, your only set. Here's your factory floor out here in the light blue. Someone knocks on the door. I need some instructions. Says, all right, hold up. Let me write this down. Transcribes it onto a piece of paper, i.e. the strand of RNA. That then you slide it through the door, slide it through the window of your library, give it to the construction worker. Construction worker takes it out, reads those instructions, and builds a protein. So again, my recommendation when you're studying this is study this easy stuff first, the big picture. Then you should add in details. So definitely know that transcription goes from RNA to translation. Notice, too, that this arrow does not go that way, right? Because without some exceptions that we'll talk about later, RNA does not turn into DNA. Therefore, when you get an mRNA vaccine, it is not going to mess with your genome of your DNA because it's RNA and it does not go backwards with some exceptions, and we'll talk about those exceptions. So any questions about this slide right here? All right. Again, this chapter more than any, I recommend downloading the PowerPoint so you can watch these videos or finding these videos on YouTube. But yeah, I would watch that. It's the same thing I'm going to show you, but it's animated, and it really helps to see it animated, I think. So here we go. What is the language of nucleic acids? I've already said this. This is why I started off with my analogy before we jumped into it. What is the language of nucleic acids? As we already said, it is the DNA. That's the language. It's the sequence of nucleotide bases. So in my factory analogy, the language was English, right? It was just those letters of the alphabet. You put them together to make words, right? And then those words can be translated. Again, bring this piece of lumber and attach it to this piece of lumber using this bolt, right? It's words, English words that you translate into a three-dimensional object. In this case, again, it's the sequence of nucleotide bases. Your book also points out, not that you need to know this, so I'm going to exit out. A typical gene contains thousands of nucleotides. You don't need to know that. As you already know, DNA is transcribed into RNA. And here's what you didn't know necessarily, but the bases of the RNA are complementary to the DNA strand. And that will be important for at least a couple of questions on your exam, because I might give you something like this. Like, here's a strand of DNA. A, G, G, T, C, A. Right? There's a strand of DNA. And the question might say, transcribe that. So the first thing, and that's it, right? The first thing you need to know when I say transcribe that is that you're going from DNA to RNA, right? So there's your DNA. What you want to do is RNA. And now, like I just said, the bases are complementary. What do I mean by that? Well, does anybody remember what goes with an A?
C.
Good, and you'll need to know that. Anybody remember what goes with a G?
C.
Good. Anybody remembers what goes with a T? I gave it away. Yep. How about a C?
G.
Good. And how about an A?
T.
All right. Now that's almost done, but it's incorrect. And this is the mistake people will make on the multiple choice exam, because that will be one of the choices. Problem is, we're going from DNA to RNA. And remember, RNA does not have T's. It only has U's. So when you get the question to transcribe this strand of DNA, then your answer will actually be UCCAGU. And yes, there will be at least one question like that where I ask you to transcribe something. And if you're trying to be quick, multiple choice, you want to eliminate some options, anything that has a T in it is not going to be the answer. If it says transcribe it and it has a T in it, that's not the answer, right? Because RNA does not have a T. Any questions about that?
Yes.
Yes.
DNA sequence of nucleotide bases. That's really these things.
Yep.
Right here.
Yes. So this bullet point right here is this right here. The DNA sequence of nucleotide bases. That's the A's, the C's, the G's, and the T's. Or in your book of instructions, it says A's, A through Z's, right? All those letters. All right. So any other questions? Good question. All right. I see some notes being taken. So the next word for the online people or for attendance is mud. Like that thing you get when you mix dirt and water, mud. And here's a hint. This is the only time when all four words are going to actually go together and make sense. Might not make sense to all of you, but they do make sense. Anyway, let's talk about translation, because we're done talking about for now we're done talking about transcription, because again, this is just an overview. Translation. It's a conversion, right? Just like any translation is. If you're a translator, usually that means you're translating from one language to the other. If you watch these COVID briefings from the state government, you've got somebody speaking English, and then you've got Julie Berry out there doing sign language, right? She's translating it from English, from spoken English to American Sign Language. That's translation, one language to the other. And in this case, you are doing the same thing. You're going from one language to the other. In this case, you're going from the nucleic acid language to a polypeptide language. Just like in my analogy, you were going from the written words of English to the language of a three-dimensional object, whatever it was that you were building, whether it be lumber or metal parts or electronic parts, whatever. So in this case, in this new language, the polypeptide language, remember the last language we're talking about, the nucleic acid language, it's the A's, the T's, the C's, and the G's, and the U's if we're talking about RNA, right? That's that language. The polypeptide language, right? We're talking about polymers here. We already know from previous chapters that those monomers are amino acids, right? So this is the language. It's the amino acid, right? So we're going from DNA and RNA, right? We're going from these A's and C's and T's to G's to amino acids. How does that work? It is the sequence of the RNA bases now, right? Because we're done with DNA. We've already transcribed. So it's the sequence of the RNA bases that dictates the sequence of the amino acids of the polypeptide. Just like I was using in the, like I was saying in the example. You have this set of instructions written in English, right? A's through Z, A, B, C, D, E, F, G, H, I, J, K, L, M, N, P, Q, R, S, T, U, V, W, X, Y, Z, right? That's the language. And those different words, those letters are put together in different combinations to make words. And those words instruct you. Paint this part red, paint this part blue, right? It's instructions and you just follow it. Same here. The RNA bases, those A's, C's, U's, and G's say, bring in this amino acid. Good. Now attach this amino acid to it. Good. Now attach that amino acid. That's it. That's how simple it is. Those A's and C's and T's and G's in the DNA, we just copy that down as A's and C's and U's and G's and RNA. And then we take those letters actually do form words, and those words say, bring me this amino acid, bring me that amino acid, bring me amino acid X, bring me amino acid Y. So it's the same situation here. We have letters that form words, and those words say, this is the next step in building this three-dimensional thing. In this case, the three-dimensional thing will be a protein. Whew. So the next slide I'm going to show you is basically a picture of everything I have on this slide. Here we zoomed into a strand of DNA. Obviously, it's going to be much longer than this in real life, but we zoomed in. We're looking a little bit closer, and we happen to have 3 genes that we can see. So now we're zooming into gene number 3, not even the whole thing, just this little bitty section of it. And here in gene number 3, you can see the DNA strand, the code, the letters, AAA, CCG, GCA, AAA, right? Just letters. That's just an example. Certainly don't memorize that. So if you were to transcribe that, like I said earlier, and you'll have to do that for the exam, you're going to have to do the complementary for the RNA, except remembering that RNA doesn't have T's, it has U's. So again, T is complementary to A, but we don't have that, so it's a U, hence those U's. G is complementary to C. C is complementary to G. Again, T is complementary to A's, but we have U's instead. So that's it. That's transcription. And now, here's what I'm saying. Those letters, just like in how we operate, those letters come together to form words. But it's easier in this case than the English language. Let me ask you this. How many letters is the average English word? Oh, I don't know. Is it? I really don't know. But for sure, it's a wide variety, right? Because you go from a letter like A, or a word that like A, to a word like supercalifragilisticexpialidocious, right? So there's a big variety. Here in this language, nice and simple. Every word, and I'm going to talk about this in more detail later, it consists of 3 letters. But anyway, the point is, again, we have these letters. These letters come together to make words. And these words, all they do is tell you which amino acid to bring. So this word, which we call a codon, and we'll talk about that later. All these words are called codons. This word says, bring me a square amino acid. This word says, bring me a circle amino acid. This word says bring me a triangle amino acid. And this word says bring me a square amino acid. Now keep in mind, this is just an example. So there are no square, circle, triangle amino acids. It's just to simplify things. And again, we'll talk about this word codon here in a second. So any questions so far? All right. So the flow of information that starts with the gene in your DNA and goes all the way to the protein. I've already said this, but now I'm saying it in the way your book presents it. This information is based on a triplet code. It's another way of saying what I've already said, that you've got these letters and they make up words, just like they do in the English language, except here, the words are always 3 letters long. So that's what we mean by triplet code. The instructions for the amino acid sequence, right? Like bring me the triangle, bring me the circle, bring me the square, all that. Those instructions are written in the DNA, as we already know. That's then transcribed, it's copied onto the RNA. And these are words. And here's something you do need to know. And we talked about it on the last slide, and I told you we would talk about it in detail. Here we go. Those words are called codons. So if you're taking notes, you could almost write down codons equals words. If you're thinking about things using my analogy, then codons equals words, right? The DNA sequences, those are the letters. Letters always make words. And in this case, the words just, all they do is tell you what amino acid to bring. I should have brought my kid's little toy. That would have been perfect. I meant to do it. I forgot. You ever seen those like baby toys? They're like a long string of things and they're different shapes and you like kind of plop them into each other. That would be like that, right? You know, say, bring me the orange rectangular one. Okay. Now bring me the red smooth one and put it in there, right? And that's all you're doing. You're just making a chain of things. And it's these codons that tell you which ones to bring. As you already know, this is being very repetitive. It doesn't matter. Somebody's still going to miss this on the exam, even though I've said it a million times. The codons in the DNA, right? So those 3-letter words in the DNA are transcribed in the complementary codons in the RNA. So we've already said that. Except now we're not just saying A's and C's and T's and G's. We're getting more specific and saying these 3 letters in the DNA are transcribed into these 3 letters in the RNA. And then these 3 letters in the RNA are translated into a single amino acid, right? That's it. That's how it works. It's a language. So and in this language, these words have 3 letters. Each word says, bring me a specific amino acid. And then again, you just, like a chain, this thing is attached to this thing, is attached to this thing, is attached to this thing. It's a long chain of amino acids. Are there any questions so far? The next slide shouldn't be there because we've already seen it. So I'm going to skip past it. So let's talk a little bit more about this genetic code. Now you know some good background knowledge and some words, or we can talk a little bit more about the genetic code and you'll know what I'm saying. Now, of this bullet point, this big second one right here, of this main bullet point, this is probably one of the less important ones, especially right now, considering that we're in the middle of a pandemic. You might argue this one's a little bit less important, but to me, it's one of the more fun ones, but kind of a nerd that way. Anyway, the next slide I'm going to show you has a vocabulary word. You don't need to memorize it. I'm not going to ask you about it. I probably won't use it again. But the genetic code, it's a set of rules that converts RNA sequences to amino acid sequence. That part of you don't need to know, right? You don't worry about that. But this other stuff, well, let's talk about it. Does anybody know how many words there are in the English language?
Like 25,000 or something.
I don't know that one either, but okay, I'll take your word for it. I don't know that. What I do know is that in this language, there's only 64 words. Of course, we call them triplets, right? You call them 64 triplets. You call them 64 codons. You call them 64 words. I don't care. The point is there's 64 of them. You don't need to know that. You don't need to know the number, but I'm just letting you know. Out of the high number of words there are in the English language, that's complicated. But here, there's only 64 words. And of course, the English language too, for the letters that make up those words, you go from A to Z, right? You have all those different combinations. This is a very simple language. There's only 4. Or if you're into computers and things like that, it's like just a little bit more complicated than that. You know, in computers, you might have binary codes, 0s and 1s. Here you have A's, C's, T's, and G's. if independent work wasn't already over. There is some really fascinating work going on where people are working with DNA to store information, like you would a solid state hard drive or something like that. Anyway, of these 64 words in this language, 61 of them actually code for an amino acid, meaning 61 of those words says, bring me this amino acid, bring me that amino acid, bring me amino acid X, bring me amino acid Y, whatever the case may be. And three of those 60 words just say, all right, we're done. Quit building stuff. You're done. It'd almost be, again, if you're reading that set of instructions, if at the end, it literally said the end, which wouldn't make much sense in my analogy because a human would just recognize the fact like, oh, those are the instructions are done, so we're done. But in this analogy, we need that. Right? This isn't an analogy. This is all real life. So again, this part you don't need to know for the exam either. You don't need to know the numbers. You don't need to know that there's 64 words, 3 of which say stop. Just know, though, that most of them say, bring me this amino acid, bring me that amino acid, and a very small amount of them say stop. Stop doing what you're doing. Another thing, and this is kind of intuitive. I haven't said this specifically, but it's kind of been hinted at. Any given RNA triplet, i.e. any word, any codon, however you want to look at it, always specifies a given amino acid. Meaning, in my body, if you were to look at my genes and you see my RNA, my mRNA, I'll talk about that later, you see the code GAC. That is going to code for a specific amino acid for me. I had to memorize these. I don't have them memorized anymore. But whatever amino acid is for me, it's also for you. Maybe it's leucine. So for me, GAC means bring me a leucine. For Thornabar, it means bring me a leucine. For the plant you gave me, that means GAC. And its codon, that means bring me a leucine, right? It's all the same, whether talking about humans, plants, bacteria. It's universal for all life that we know. One DNA triplet, one word, always equals a specific amino acid. And that's what this next slide shows you. And you might have to use this on the exam. Most likely, if so, it will be extra credit. I already told you that I was going to ask you to transcribe something, right? I told you I was going to give you a strand of DNA and ask you to transcribe it to a strand of RNA. And then the next step from that is I might ask you to translate that strand of RNA to a polypeptide. And here's how that's going to work. Someone give me some random, well, I'll just do it. This will be quicker. What did I say last time? GAC. And now because I like football, specifically the Knowles, which are not doing good this year, I'm going to say ACC in honor of them. All right. So here's my first codon, my first word, my first triplet. It's GAC. So here you start in the middle. Here's the G. All right. Now I need an A. Oh, there's an A. All right, now I need a C. Here we go. And that brings me to ASP, aspartic acid, right? So if I were to translate that, I would abbreviate, put ASP, because that word, that triplet, that codon says, all right, well, we need that amino acid on the chain. And we need that amino acid to be covalently bonded to the next amino acid, which is ACC. So let me start again. All right, A. Okay, C, and C. Threonine is my next amino acid, and I would abbreviate that the same way it's abbreviated here, THR. There we go. And that's how it works. And that's what you might have to do on the exam, and it would be extra credit. So, any questions about that? You certainly don't have to memorize any of this. Again, you might have to use it once for extra credit. If you guys are biology majors, I would almost definitely have this on the exam, and it would almost definitely not be extra credit because that would be something you would use later on, but you probably won't. Your study guides, if I remember correctly, do require you to memorize this, the stop codons. U, A, U, A, G, U, G, A. Are they still number one? Speaking of football, is the Bulldogs still number one? Or are they, I should say? Anyway, in your study guide, I think they do require you to memorize it. So yeah, do it for the study guide, but don't worry about it for the exam. Like I've already said, and here's where it gets interesting to me. The genetic code is universal. So if it says, if I have a code in my body that says, bring me a tryptophan, it would be the same in all of your bodies. And it would be the same in the plants living in my house and the cats that piss on my furniture from my neighbors, right? Every single living thing has the same code, which actually makes it very interesting and also very useful, because one species can produce protein from another. So if you know anybody who's taking insulin or human growth hormone, that was a gene that was taken out of a human cell and transplanted into a bacteria, and then the bacteria just makes the protein. It doesn't know any better. It doesn't say, that's a human protein. I'm not making that. They don't do that. It's universal, so it's made. And bacteria are just great little factories for it because they don't require much. They multiply quickly, right? You don't want to have like an animal doing it because it just takes too long. That used to be how we used to get insulin. But anyway, it also allows scientists to mix and match genes from species. This is what I'm really fascinated in as the nerd that I am, because I like and also I should tell you that my thesis work was on this kind of stuff, genetics. So I would love if I had the money and the capability, the lab equipment, to take the genes responsible for capsaicin, which the things that make peppers hot, and splice them into other fruits and like tomatoes. Have yourself a spicy tomato. I don't know. I think that'd be cool, and really up your salsa game. Anyway, again, the big thing from this slide you need to know is the genetic code is universal. There are exceptions to that, but we won't get into it. Here's what I'm talking about. This is some pictures from your book. Can anybody tell what's up with these pigs and with that mouse, I think, that baby rodent, whatever it is? Yeah. What's up with them? How are they different than most?
They've been genetically.
You got it. They've been genetically modified. What happened? This is a real life example. What they've done is they've taken genes from jellyfish, if I remember correctly. I wish I could remember the name of the gene. Luciferase, something like that. Anyway, it's a gene that causes them to glow. So they put that into these animals, and the animals literally glow. It doesn't hurt them any other way. They just produce this extra proteins, what causes them to grow. This one down on the bottom left, this is not from your book. This is not from real life either. Someone was hypothetically saying, what if we took those genes and put them into trees so the trees glowed? That way we wouldn't have to have street lamps, which I thought was great in one sense, because obviously you would have a lot less electricity. But that would be absolutely catastrophic for the environment and the ecosystem. And we'll talk about that in later chapters. But when we get back on Monday, we will actually we're done with the overview, right? So next, we're going to go into detail how we go from DNA to RNA and RNA to DNA. And the last word for those of you online is football. Football. So basically all 4 words. It's a song and the artist who does the song. Any questions from anybody online? I'll be on office hours if you need me. I'm going to stop recording. You guys have a great weekend. Don't eat too much candy if you're trick-or-treating or having it.
All right. That's pretty good.
This is really fascinating stuff. This is my second favorite part of the